Hi, hello all the NEET aspirants. Today we are here to discuss about the topic plant growth and development. So, the first segment or the first part of this chapter includes growth, development, phases of growth and different growth rates, different growth patterns and different developmental pathways that we have to learn with respect to our syllabus. Fine. So, if you look into growth, how can you define growth? Growth is defined as in a plant, okay. Growth is defined as irreversible, permanent increase, in shape and size of an organism. Irreversible or permanent increase in the shape and size of an organism is said as growth. We have learned this in living world also, right? So, yes, growth in plants is called as open form of growth. Growth in plants is called as open form of growth. This is because in plants there are two kinds of meristems. One is primary meristems which help in the or which build the primary plant body, and second are secondary meristems which increase the girth and size of the plant, whereby plant will show indeterminate growth. Plant will show what ma? Indeterminate growth. Where growth will be throughout their life. Growth is seen. This is because of what? Primary and secondary meristems. Primary meristems and secondary meristems. During growth in a plant, the rate of anabolism during growth, if you see the metabolic activity, during growth, if you consider anabolism or anabolic reactions, anabolism is greater than catabolism. Clear? If you see the metabolic activity during the growth of a plant, anabolism is more than the catabolism. So, these are the primary points that you have to remember. Where the irreversible permanent increase is said as growth, in plants you will see open form of growth, that is also called as indeterminate growth pattern. This is because of primary and secondary meristems. So, yes, and if you look into the metabolic activity, anabolism is more than catabolism. If you look into the apical meristems here, apical meristems, be it shoot apical meristem or root apical meristem. Apical meristems in a plant, be it shoot apical meristem or root apical meristem. Along with one more type of apical meristems, what are they? Intercalary meristems. Okay, shoot apical, root apical and intercalary meristems which are present in between two nodal regions, which are present in the mature tissues in between two nodal regions. Okay. So, yes, all these type of primary meristems will increase the length of the plant. They will increase the length of plant body. Their primary activity is elongation. Elongation. Clear? Next, when it comes to vascular cambium in the stele region or cork cambium in the extrastellar region or cortical region. Okay? Vascular cambium in the stellar region, two secondary meristems are present, right? What are the secondary meristems that you see, ma? One is cork cambium, we have learnt in anatomy, right? Cork cambium and vascular cambium. Vascular cambium will conduct stellar secondary growth. Vascular cambium will conduct what ma? Stellar secondary growth. Whereas cork cambium will conduct secondary growth in cortical region. Cortical region. Their main activity is to increase the girth or size of the plant. Increase of girth or size of the plant body. Clear? This is what you have to observe mainly. Okay, why plant is showing indeterminate growth? That is because of primary meristems and secondary meristems. Primary meristem's main activity is to increase the length of the plant or to conduct elongation. Secondary meristems, be it cork cambium or vascular cambium, their main activity is to increase the girth of the plant, girth or size of the plant. Then if you look into the growth in plants, is growth in plants measurable? Is growth measurable? Yes, it is measurable in many parameters. You can measure growth. You can measure growth as increase in the cell size, where a single watermelon cell can increase up to 3,50,000 times. And by increase in volume, if you look into fruits, storage organs, vegetables, they increase in their volume and thereby growth you can measure in the fruits and vegetables or storage organs. Increase in surface area, if you can see leaves, young leaves will have less surface area and matured leaves will have more surface area. Thereby you can measure the growth in leaves by surface area. And then increase in fresh or dry weight, this also you can use as a parameter to measure the growth in fruits, vegetables and storage organs. And increase in cell number, if you consider a maize cell, okay, maize cell, root apical meristem of a maize cell, wherein 17,500 new cells are formed every hour. Okay, every hour 17,500 new cells are formed. And then if you see bacterial culture, it might be algae or yeast, they also increase the cell number, they also show their growth in the parameter of what ma? Increase in cell number. Okay, these are different parameters where we can measure the growth or estimate the growth. Then next it comes with phases of growth. Okay, there are three phases of growth. First is meristematic phase, second it is elongation phase and third is maturation phase. If you look into first, that is meristematic phase. Meristematic phase. How cells will be in the meristematic phase? Cell size is small. Okay, cell size is small. And you can see a large nucleus, cell size is small, a large nucleus you can see, okay, dense cytoplasm and cell wall is thin, with plasmodesmatal connections, with what ma? Plasmodesmatal connections. Okay, clear? Cell wall is thin with plasmodesmatal connections. What are the plasmodesmatal connections? Those connections which connect the cytoplasm of two different cells, right? Those connections which connect the cytoplasm of two different cells, those which traverse along the cell wall. Okay, cell wall is thin, then cytoplasm, large nucleus, cell size is small and plasmodesmatal connections are seen, and there are no intercellular spaces, no intercellular spaces in between the cells which are in the meristematic phase. Second is the elongation phase. If you see how cells are in elongation phase, yeah. In the elongation phase, if you consider elongation phase, cell size increases. How cell size is increasing? Of course, we know from Cell the Unit of Life we have learnt that vacuole occupies 90% of the cell volume. Increase in vacuolarization happens in the elongation phase. Increase in Vacuolarization, okay, increase in vacuolarization, thereby cell size increases, and cell wall depositions are seen. Cell wall depositions are seen in the elongation phase. In elongation phase, what changes you observe beta? Increase in vacuolarization. As vacuole size is increasing inside the cell, cell size also increases, and cell wall deposition is seen in the elongation phase. Then comes the last phase, that is maturation phase. What is the third phase beta? It is maturation phase. Third is maturation phase. In maturation phase, all the cells will attain structural and functional differentiation. All the cells will attain structural and functional differentiation. Clear? These are the modifications of cells or the patterns of cells which are observed in different phases of plant growth. Okay, this you have to keep it in your mind, ma, very important. Yeah, then comes about growth rate. Okay, growth rate. What is growth rate priorly? Growth rate is nothing but increase in the growth per unit time. What is growth rate, ma? Growth rate is nothing but increase in the growth of a plant per unit time. Okay, increase in growth of any organ or plant per unit time is called as growth rate. Two types of growth rates are observed. One is arithmetic growth and the second is geometric growth. In arithmetic growth, what happens is a single cell dividing into two cells. Of those two daughter cell, one gets permanent and the other will show meristematic activity. Again, when this cell divides into two, one cell gets permanent and the other shows meristematic activity. In the arithmetic growth rate, you have to remember that only one of the daughter cell is conducting the meristematic activity, the rest daughter cell or the other daughter cell is getting permanent. Okay, this is mainly used in learning, you know, root elongation in the plant, root elongation, where arithmetic growth is followed. Okay, if arithmetic growth rate is predicted in a graph, on x axis time is taken, on y axis height of the plant is given. In that case, you can see a linear curve. Okay, you can observe a linear curve. Okay, the formula to calculate arithmetic growth rate from this graph is L1 is equal to L0 plus RT, where L1 is equal to final length, L0 is initial length and R is growth rate and T is time. Clear? This is about arithmetic growth rate which is showing linear curve. Clear, ma? This is showing what? Linear curve, and the formula used to calculate is L1 is equal to L0 plus RT. Then comes geometric growth rate. Geometric growth rate is mostly seen in the bacterial cultures, bacterial or microbial cultures, wherein geometric growth rate, one cell divides into two, one cell divides into two, each daughter cell is giving rise to two other daughter cells. Here there is no concept of a cell getting permanent. Okay, each daughter cell will give rise to two more daughter cells. This is how they increase in their cell number. When this pattern of growth is depicted on a graph taking time on the x axis and size of or weight of the organ on y axis, you will see a sigmoid curve. Or S-shaped curve, or what, ma? S-shaped curve, wherein 3 phases of growth are seen. The first phase is lag phase, second is log or exponential phase, and third is stationary phase. In the lag phase, during the initial phase of growth, cell number is less. Okay? During the initial phase of growth, during initial phase of growth, during initial phase of growth, cell number is less. Cell number is less. Then during log phase or exponential phase, gaining up all the nutrients and increasing in the cell number is seen rapidly or drastically. This you will observe in the log phase. And due to limitation or decrease in the cell, due to decrease in the space, due to decrease in the space in the culture, okay, due to decrease in the space and nutrients and due to increase in the toxins, decrease in the space and nutrients, increase in the toxins, again they attain a stationary phase. Okay. So, here 3 phases are seen. What are they beta? Lag phase, during initial phase of growth cell number is less, then log phase or exponential phase where rapid or drastic growth is seen, and then stationary phase is observed. This is because decrease in the space or nutrients or increase in the amount of toxins that are released by the bacteria in the culture. Right. So, this is about arithmetic growth and geometric growth, and the formula that is used to calculate the geometric growth rate from this graph is W1 is equal to W0 ERT, okay, where W1 is the final weight, W0 is the initial weight, okay, initial weight, and E is the base of the log, R is the growth rate, and T is the time. This is how geometric growth rate is observed. This is the arithmetic growth rate. Yes. Now, for the exam point of view, what you have to remember? You should know what is arithmetic and what is geometric growth rate. You should know the graphs. You should know what is there on the x axis and y axis. What is the shape of the curves that you see in the graph, and what is the formulae that is used for arithmetic growth rate and geometric growth rate. This is what you have to know. Fine. Then here you can clearly see when zygote is divided, it divides in all the planes. As you know, zygote is totipotent, right? It is totipotent, and it divides in all the planes to give rise to an embryo or to give rise to a mature cells, okay, group of cells. Like, you know, for the formation of embryo, the cell is dividing in the all planes, which shows a geometric phase of growth. And once a tissue is formed, you can see arithmetic phase of growth where few cells are getting differentiated for the organ formation. From the zygote, if you can see up to the formation of embryo, geometric phase of growth is seen as it divides in all the planes, and after certain amount of tissue is formed, then you can see the arithmetic phase of growth, because organ differentiation has to take place. So, here from this diagram you can clearly see the geometric phase and arithmetic phase of the growth that takes place in plants. So, if you can see, this is the shoot apical and root apical meristematic region where arithmetic phase of growth is observed at its apex. Okay. Then measurement of growth. Growth is measured in 2 ways. One is absolute growth rate. What is it, ma? Absolute growth rate, and the second is relative growth rate. Absolute growth rate, and the second is relative growth rate. Relative growth rate. Okay. Absolute growth rate is calculated by taking the final size minus initial size. Final size minus initial size will give you the absolute growth rate of a particular organ. If you consider it for a leaf, this absolute growth rate if you consider, where 10 centimeter square is the final size of this leaf and 5 is the initial. 10 minus 5, so the absolute growth rate is 5 centimeter square for this leaf. Okay. How you will calculate the relative growth rate? Relative growth rate can be calculated by taking final size minus initial size, final size minus initial size by initial size into 100, by initial size into 100. Okay. Final size minus initial size by initial size into 100. So, both this absolute growth rate and relative growth rate are quantitative comparisons of growth. They are what ma? Quantitative comparisons of growth. Quantitative comparison of growth, wherein in relative growth rate if you see final size minus initial size. What is the final size here beta? 55 minus initial size is 50 by initial size into 100. If you calculate, you can get 10 centimeter square is the relative growth rate that is seen. Fine. Both the formulae are important ma. You may get numerical questions using this formula. Okay. Then, yeah, this is most important, differentiation, dedifferentiation and redifferentiation, where the questions are asked in connection with the chapter anatomy. Okay. Anatomy. Fine. So, if you see ma, clearly if you observe, what are the meristematic tissues here in the plant for the formation of primary plant body? What are the meristematic tissues? Two meristematic tissues are there. What are they? Meristematic tissues are of two types. One is apical meristems and second is intercalary meristems. Intercalary meristems. Apical meristems are of two types. What are they? Shoot apical meristem and root apical meristem. So, this meristems will form the primary plant body where few cells or tissues are differentiated. Okay. Those differentiated cells means what ma? They lost the capacity of division. Okay, and they has attained structural and functional maturity. So, what are those cells called as? Differentiated cells. Differentiated cells form up to be differentiated tissues. Okay. So, as a result of the differentiation, what are the cells or tissues that are formed? Simple permanent tissues, complex permanent tissues. Okay. What are simple permanent tissues that we have learned? Parenchyma, collenchyma, sclerenchyma, right? Parenchyma, collenchyma and sclerenchyma. Fine. Complex permanent tissues include xylem and phloem. Yes, these tissues, these differentiated tissues will form up certain regions of the plant body where you will call it as differentiated regions, like you know cortex, endodermis, pericycle, medulla. All these are what? Differentiated tissues or differentiated regions. You have to remember, they do not have any capacity of difference, they do not have any capacity of meristematic activity. Okay? So, these are what ma? Differentiated tissues. Now, few of these cells will regain or re-attain the capacity of meristematic activity. What are those called as? De-differentiated tissues. As they lost the power of differentiation, they are regaining the capacity of meristematic activity. They are called as de-differentiated tissues. De-differentiated tissues. If you look into the de-differentiated tissues, where you can see what are the de-differentiated tissues, in the stellar region if you see, you can find vascular cambium. In the stellar region if you can observe here, you can see vascular cambium. Vascular cambium. In the cortical region, if you observe, you can see cork cambium. In the cortical region, you can see cork cambium. Okay. In the stellar region, you can see vascular cambium. In the cortical region, you can observe cork cambium. Then, due to the activity of this meristematic, these secondary meristems, due to the activity of secondary meristems, few differentiated tissues are formed again. Those are called as redifferentiated tissues. Redifferentiated tissues. Tissues. What are they? What are they, ma? Cork or phellum, secondary cortex or phellogen, and secondary xylem, secondary phloem, secondary medullary rays. Secondary medullary rays. So, yes, these cells are formed due to the activity of secondary meristems or dedifferentiated tissues. So, these are called as what, ma? Redifferentiated tissues. So, this is the whole concept of differentiation, dedifferentiation and redifferentiation. These tissues you have to remember for sure what tissues or what regions comes under differentiated tissues, what comes under dedifferentiated tissues. Okay, secondary meristems are dedifferentiated tissues, and then what tissues or what regions comes under redifferentiated tissues. This and all you have to fix it in your mind. Many a times they have asked you questions in the exam. Okay, so this is very important, ma. Then comes development. Development is nothing but the activity of growth and differentiation. Growth and differentiation. Sequential changes that are observed in a plant is called as development. The sequential changes that are observed from the juvenile phase or seedling phase to the maturation phase is called as developmental aspect. So, if you see the seedling stage, first is what, ma? Seedling stage, and then juvenile stage, then comes the maturation stage, and flowering, seed formation, and senescence or death. These are different sequential changes that are observed in the developmental process of a plant. So, here in the development you will learn two phenomenal concepts. What are they? Plasticity and heterophylly. Plasticity. What is plasticity, ma? What is plasticity? Plasticity is nothing but the plant following different developmental pathways in response to its phases of growth or the environment is called as plasticity. Plant following, okay, different developmental pathways either in response to phases of growth, or in response to the environment is called as plasticity. One of the best example of plasticity is heterophylly. One of the best example of plasticity is heterophylly. Heterophylly means what, ma? Different shapes of the leaves are observed in the same plant. Different shapes of the leaves are observed in the same plant. One in juvenile phase and the other in maturation phase. Best examples for heterophylly are cotton, coriander and larkspur. One more example that is shown for heterophily, wherein different leaf structures are shown by same type of plant in two different habitats, One is in terrestrial habitat and the other is in water habitat. The best example is buttercup. Okay. So, yes, with this we have completed the first phase of the plant growth and development. In the coming up session, we are going to discuss about plant growth regulators, which is most crucial for your exam. So, stay tuned and thank you.